Great. So let's pick up from where we left off yesterday. So we finished the analysis. We performed our UMAP, our two-dimensional clustering. And the next step is the identification of marker genes. So we're going to start by going through a little bit of theory about what these marker genes are. Before I start, is there any question from any of the material that we've covered so far? Great. So like I mentioned before, the next step is the identification of marker genes. And so first we'll start by defining what is exactly a marker genes. So the marker genes are those that give each cell cluster an identity, because what we've used so far is that based on the gene expression profile for these different cells, we're trying to group them together based on those that have similar expression patterns. However, what distinguishes one cluster from another? What can we say about one cluster of cells that we cannot say about the other one? And the answer to that is that certain clusters will have certain genes that are expressed either much higher or much lower compared to the other clusters. And because expression of this gene is unique in this cell cluster, then we'll call this particular gene a marker gene. So this second bullet point just very quickly reiterates what I mentioned, is a marker gene is that is differentially expressed in one cell population compared to the rest. And the identification of cluster genes is done using an analysis that is called differential gene expression. Now, differential gene expression is a pretty broad topic also, so it's statistically challenging to say for sure that a certain gene is expressed differentially across two conditions or across two, in this case, cell populations. And you're probably heard of many methods or many statistical approaches that people came up with in order to deal with this issue. In the case of Surat, which is the library, the package that we're using for our analysis, the way it does it is using a Wilcox rank-sum test. So before we move on to the code, I just wanted to make a quick side note also regarding differential gene expression. Like I mentioned before, this is a challenging problem. And the reason why it's so challenging is that it's hard to model the variance for gene expression. What does this mean? So before I show this plot, I use this figure to explain how to identify the highly variable genes that were these pink dots. But what this plot is also showing you is that even though they were able to model the relationship between variance and average with this trend line over here, you can see that clearly it does not cover all of the data points. It doesn't do justice to the real spread of the data that is present. And so in the effort of trying to quantify for a gene, say we have an average normalized recount of 10, how much variance do I expect? So if this same gene has an average expression of 20 in another condition or in another cell, is that difference of 10 significant? How can we figure that out? And there have been many methods trying to deal with this issue. I believe there were a few people that they said they worked with RNA sequencing before. So maybe we can do a quick show of hands of these people and whether you have used and whether you have performed differential gene expression before. Right. Yeah, we have 3 people. Oops. Right. So you've probably used certain packages such as D-seq or HR or something like that. And the only point I want to make here is that there are packages that, because it's so challenging, there are specialized packages for this task. And usually they try to overcome the fact that there are so few replicates, right? Because if you had multiple replicates for the same condition or for the same cell, then this becomes an easier problem. And the point I'm trying to make is that with Surat, it's actually not performing a very sophisticated method for differential gene expression. It's using a very common statistics test, the Wilcox rank sum, which just evaluates the difference in mean between 2 distributions. And the reason why we can this package can afford such a relatively simple test is because if you cluster a cell in a cell population or a cell cluster, this cluster contains many cells, right? It can contain up to a few hundred, well, depending how many cells you started with. It could, in our case, we started with over 2,000 cells. Each cluster easily contains a few hundred cells. So we're considering these few hundred cells as replicates of the same type of, as replicates of the same condition. So having that many replicates, there is no reason why we can perform a more straightforward test. Okay, so let's switch back to our R code. Nope, it's not Kahoot time yet. Hopefully you guys are still seeing my entire screen. Can we do a show of hands if you are all seeing my RStudio? Right, okay, good, good. So yesterday we left off, I believe at line 220. Please somebody correct me, but I believe this is the last command that we run. So just to bring everybody up to speed, we're going to give you a few minutes to make sure you run the code up to this point. And actually, also you can skip the jackstraw step, which is the step where we determine the number of PCs. So in line 186, this step over here, this one takes a long time. And so I would just suggest you you run it, say maybe 10 times only. So that step runs a little faster. And yeah, we're going to wait 1 minute and then we're going to continue from this step. Yeah, also please be mindful about your microphones, because We can hear some background noise. Okay, I think that should be enough. I think the entire code runs pretty quick. So hopefully everybody has a plot that looks something like this. And I don't know about you, but I really find this type of plots beautiful. And just being able to see the different cells and the different clusters. Anyway, moving on, the next cell in our code is this function called save RDS. This is not part of the analysis. It's just to save, your progress, kind of. So next time you can pick up from this point. You don't need to run everything else. Sure, probably ended with this last time. So, oh yeah, so you'll need to change this path also. You'll need to change the path to the directory where you want to save this object. So moving on, now we get to the part of finding differentially expressed features. There was one question from one student asking if we're going to go through differential gene expression analysis. And this is a step where we perform differential gene expression in order to identify our marker genes. As you can see, it's just one line of code. So we're not going to go through too much detail. We're going to look at the output. So you know what numbers we're getting, what is going on. But yeah, other than that, we cannot really expand too much on the topic of differential gene expression. So let's move on to this cell over here in line 232. The first few lines is really just testing, or really just finding clusters marker genes for individual clusters. So for example, if you see the first line of code, the function find markers is going to perform the differential gene expression, and is going to find genes, marker genes that are differentially expressed in only cluster number 2. So if you remember our UMAP plot, cluster number 2 is here, is colored with this sort of dark green. I believe it's this one over here. And this parameter over here is just the requirement because the gene could be differential because it's not expressed at all in other clusters, right? So you can set up some minimum requirement. You expect at least 25%. You expect this gene to be present in at least 25% of the other clusters. So you can make it testable. And the second line of code, well, the third line of code actually in line 237 is performing pretty much the same function for cluster number 5, but instead of comparing it to everything else, is comparing it to clusters 0 and 3. And finally, this last function is finding all of the markers for all of our clusters. And it's also setting some minimum cutoff. We're going to see what this parameter means. So you can go ahead and run this cell. Should take a few seconds. Yes, there is a question from Brian. Brian, do you have a question? Oh, you wrote it down. Okay. Is there a reason why line 226 did not run? Oh yeah, like I said, this is to save your progress. So you just need to update this this variable with the path where you want to save the progress on your computer. So here I'm getting an error because I don't have a folder named output. So yeah, you just need to update that. Now going back to the results from our marker genes. So let's look at the output from this first line, which is find markers for cluster number 1. And here we're just showing the top 5 genes that we identified. So here we have our gene names. We have the p-value and the average log2 fold change. These parameters that derive from the differential gene expression calculation. And adjust p-value to correct for multiple testing. And these 2 parameters that indicate the prevalence of this gene in the testing groups. So this table should be sorted based on the most significant genes first. So in our case, for the way we'll understand this is that for cluster number 2, the gene IL-32 is differentially expressed, meaning that we can identify cells in cluster number 2 based on their expression of this particular gene. And because the average log2 fold change is positive, this means that this gene is overly expressed in this particular cluster. We can look at the output for the second line of code, which is the same idea. We're just looking at cluster number 5. These are our marker genes for cluster number 5. We have our p-value, and these are the parameters. Yeah, and lastly, this is the output we get when we run the function find all markers, which finds the markers for all the clusters. And the values, they are the same. They derive from the differential gene expression analysis. And here we have the name of our marker gene and the cluster for which this gene is a marker. So this last line of code, what it's doing is just selecting the top 2 for every cluster and printing that and printing those genes.
Giovanni, could you please explain more what the PCT1 and the PCT2 mean?
Yeah. So when you try to calculate differential gene expression, you want to make sure that this gene, we have values for this gene in the 2 groups that you're comparing, right? Like we saw before, if this gene has an expression value of 0, it's not necessarily a biological 0, right? It could be a missing data. So what this parameter is doing, because when you do differential gene expression, you're comparing 2 groups. And in this case, we're comparing cluster number 2 with the rest of the clusters. So what this parameter is saying that we want at least 25% of the cells, in this case, of either group to contain this gene, to have an expression value for this gene.
Okay, and, got it. Thank you.
Yeah, so that's the minimum. And here, all the values that we observe here should be greater than 0.25. Right, we can move on to the next line of code in line 244. So here we're doing yet a similar exercise, finding the clusters, the marker genes, sorry, for cluster 0. But here we're adding one more parameter to our function, which is the test use ROC. So you can go ahead and run this cell. And this table should be similar to the ones we saw before, but there's one very important difference, which is this very first column, which has this value, which is my AUC. So we can do a show of hands first. Who is familiar with the concept or the purpose of an ROC curve? Okay, we have one person. Yeah, so to quickly explain what ROC is a type of analysis, is a type of plot that you do in order to test how good your classification is. And I guess we don't need to go too much into the details for ROC, but the takeaway point is that the way you test how good the classification is based on this metric, which is the area under the curve. This area under the curve represents sort of the balance between sensitivity and specificity. So the closer this value is to 1 means the better your classification is. And in this particular example, we're testing how good are we really at identifying differentially expressed genes. And here we have our AUC value for the different genes that we identified. Now we can move on to the next cell in line 249. And here we're just going to do a little visualization exercise. So I think we can first run the code in this cell. Oops. Yeah. Okay. Hopefully you'll run it by now. It should be fairly quick. What we're doing here is we're making three different plots, 3 different figures. In the first one, we're trying to plot the expression of these 2 genes and trying to get these values in a cluster per cluster to see how it's different. So the way that looks is we have the cluster labels in our x-axis, and the y-axis is the expression label for our genes of interest. And hopefully you remember the concept of a Violin plot. It kind of shows the distribution of the data points. The wider the plot, meaning the more dots you have at that position. So in this first plot, we can see that these marker genes, MS4A1 and CD79A, they have very high expression in cluster number 3. You see that the average should be somewhere in between, somewhere here, in between 2 and 4, or in between 2 and 3 for this other gene. Is very highly expressed, whereas in the other clusters, we observe that this gene really is mostly contains very low expression value. So this is a nice visualization of what a marker gene is. And therefore, we can use these 2 genes to identify cluster number 3. This is a similar idea. It's just that before here we're plotting our normalized expression value, and here we're plotting counts directly. And here we can see also for we're testing 2 different genes. We see that they have very high expression into different clusters. For this particular example, you see that it's not always very evident, very straightforward. This particular gene has high expression in 2 clusters, but it can still serve as an identifier, because most of the time, for most applications, people use multiple markers to identify a cluster. So you examine them as a set of markers. So I guess the point that we're trying to make here is that it may not necessarily stand out in only one cluster. You may run into the case where it's expressed into 2 clusters. And lastly, and I really like this type of plot also, where here we have the outline or the shape of our 2-dimensional clustering. This should have the same shape of the UMAP plot we generated earlier. And it's showing the how your different cells cluster in 2 dimensions, only that now we're coloring individual cells based on the expression value of certain marker genes. So this is going back on the same point that some genes are more highly or lowly expressed in certain clusters compared to others. It's just showing you different ways to visualize this. And in this case, you can see very clearly how this cluster over here contains really high expression of this particular gene, whereas in the other cells, in the other clusters, generally is much lower. Of course, there could be some noise, some particular cells that do have the expression, but overall, we can see that the marker genes tend to be mostly unique to individual clusters. And for this visualization, I believe it's only 9 genes that we're looking at. You can select more genes and see where in this plot they stand out. Okay, moving on. Let's go to the this cell over here on line 256. Also, if at any point you have questions about the plots or the output that we're getting, also feel free to stop me. I believe we're going a little bit faster this time. Now, moving on to line 256, we have another chunk of code. What we're doing here is yet another type of visualization of your marker genes. This first line of code, we're selecting the top 10 genes that have the larger log2 fold change, the largest log2 fold change for each cluster, and we're getting a list called top 10 genes. And after that, we're plotting a heat map where we can see the where we're able to see the expression of these top 10 genes per cluster. And here I made a little update to the code. I just commented, I put a comment here so this part doesn't run. And this is just so that we can see again our clusters and the value for the different colors. So please go ahead and run this cell. Okay. So this cell should contain roughly 10 genes for 10 genes times the number of clusters, so 80 genes. And what we're seeing here is that we're clustering this heat map where the rows are genes and the columns are cells. And the color of each entry is the expression value. So yellow means very high and pink or purple means very low. And what we can see here is that these expression patterns, they cluster based on the cells that were present in every cluster.
Hello? Oh, so, buddy, how you doing?
There's somebody that has the
UMMC Health Center. It took me a second. I was like, what is this? I've been getting, man, I don't know if you find this, but I've been getting like probably 5 spam calls a day with my US number. It's insane. So I screen most of these calls, but I figured this one was probably.
Okay. Yeah, I had to remove them, so sorry about that. Oh, I think I removed the wrong person also. Okay, we're going to deal with that during our break. So going back to our plot, what I was saying is that here in this heat map visualization, you can see that you'll obtain for your marker genes, the expression values should really stand out compared to the rest of genes. So here you can see that you obtain a band for every cluster, and this band represents the expression of your marker genes. So in the last step of our analysis, here so far we've been talking about the clusters as cluster 0, cluster 1, cluster 2, and we know what genes stand out in every cluster, but it's not biologically meaningful to name them cluster 0 or cluster 1. So we can figure out what type of cells they are exactly based on these marker genes. So for this data set that we're analyzing, the authors already put together a list of known markers and what type of cell they are a marker for. So in our example, we have cluster ID 0. And in cluster 0, we have two marker genes. And what is known already from previous studies is that these particular genes tend to be overly expressed in naive CD4 plus T cells, and so on. So based on this previous knowledge, we can assign an identity to every cluster that we found. So we're going to do that in our next line of code. Here we have a list of our cluster IDs, so the cell type. And here we're going to, this function rename idents or identities is just simply going to assign the name of these markers based on the cell based on the marker genes. And then finally, we're just regenerating our UMAP plot, including these label names for our clusters. So you can go ahead and run this chunk of code. And you can also try to make the plot a little bit bigger. So you can change the dimensions here to 7 and 7. And once you have produced this plot, this is finally the place where we wanted to get at from the beginning. We started with a population of cells, over 2,700 of them. And we had no previous knowledge of the composition of these cells, what type of cells they were, and so on. So thanks to our clustering analysis, identification of marker genes, we can assign a label, a name to these different cell populations. So this is how researchers find how heterogeneous certain tissues or cell samples really are, based on how many different cell populations they can identify in their study. And we also mentioned that one application is the identification of new type of cell types. So the way that would come around is that you can easily imagine you have a cluster where the set of marker genes don't really match anything that was previously known in the literature, right? So once you evaluate all the clusters that were present and that they match certain expectation, you'll sometimes be left with a few other clusters that don't really match the cell types you were familiar with. And that will lead to the discovery of a new type of cell, which will later, of course, need to be validated through other experiments. Yes, there is a question, Sarah.
Yeah, sorry, I think you removed Chris Hamad by accident.
Oh, yes,
Yeah.
that's the person.
So you removed two people. Chris Hamad was silent, but I think you removed him by accident. So he tried to connect, but he's not allowed to.
Oh, that's really bad. Okay.
Thank you.
Let's see. Okay, yeah, that's my mistake. Let's take a little break now then. And in the meantime, I'll try to get Chris back.
Appreciate it.
Okay. So yeah, we're going to take a 10-minute break. Okay. Right. So let's go back to our R studio. Right. So during the break, somebody asked about this violin plots and what unit are we showing here exactly. And so I had a quick look at the Seurat documentation. The default value when you plot run a violin plot, it should be the normalized expression level. So that'll be in our case the CPM value. So the way you understand this is roughly the percentage, roughly the proportion that this gene is expressed in your whole library. Okay, hopefully that's clear. And in the second plot that we make, we change the units into row counts. Okay, and then moving on, that's actually the last step, the last steps of our tutorial. There is a few cells after that, a few chunks of code. The first one is to save the plots and I believe some of the objects that you generated. So there's really no need to save the objects as this is a small data set and it's very easy to rerun every time. But if you wish to save our PBMC object, this is pretty much the object that contains all the information about the UMAP, marker genes, PCA, and everything that we did. So you can save it into a file to be read later. And lastly, our session. So yeah, we completed our tutorial. And now it's Kahoot time. Go back to our slides. So hopefully everybody is familiar with this already. It's better if you log in from a separate device so you can see the questions on your screen. And I'm going to go ahead and generate the ID. Okay, this is the ID, so please type that to join. Okay, we have 4 players so far. Does anybody else need a bit more of time to join? See a show of hands. Okay, so it seems we have all of our players, and we're going to get started. Yeah, so this is the Kahoot from yesterday, actually, that we didn't get enough time to go through. Which of these metrics is not used for quality control? Here we have almost everybody. Let's see. Oh, it actually wasn't so clear this time. Well, maybe the question was not super clear for everybody, like which metric is not used for quality control. And we've talked about before our feature count, RNA count, and mitochondrial. This being the number of genes, this being the number of unique RNA molecules, and this being the content of mitochondrial RNA. It's true, we didn't, in our analysis, we didn't use RNA count as a metric, but I did mention that is a parameter, a metric that people would look into in order to filter. And read count, read is definitely something that's not being used. Like I mentioned before, people would prefer RNA count than read count. And this is because, like I mentioned, you can have multiple reads with the same UMI, right? So these are many reads, but it's only one molecule. So you only care about the number of RNA molecules and not really the number of reads. At least when it comes to quality control. There are exceptions, but generally people care about RNA count. Okay, let's move on to the next question. Here we have Jenny leading so far. Question number 2, what is a spike in? And this is something we mentioned a few times. And so a good is a concept necessary that's very necessary to understand a lot of the things we talked about, actually. Let's see, yes, most people got it correct. I'm happy about that. So a spiking is an RNA or a group of RNAs are introduced into the sample, into your experiment, that they can serve as controls. Great. Great, moving on to the third question. What is the main assumption made in normalization? So I guess this Kahoot is a little more challenging because this is material from some of the material from yesterday. I guess we'll see who has the better memory. Okay, this one wasn't so clear either. So the assumption that is being made in normalization is that all cells contain the same amount of RNA molecules. And like I mentioned before, because we made our normalized unit of expression is CPM, and CPM represents like the proportion, right? Like the proportion of RNA molecules that are that a certain gene is using. So with that in mind, we're assuming that across cells, the amount of RNAs is consistent. Let's move on to the next question. Okay, we have some people moving up in our scoreboard. What happens to the zeros after normalization? The zeros or dropouts? Hopefully you remember that that plot from that one study that show how the distribution changes after the normalization. Yes, so the take-home message is that after normalization, the difference between a zero and a non-zero becomes greater. And of course, that's going to affect the overall distribution. And I guess the way to more intuitively understand this is that if you remember when you look at the row count, the distribution looks very continuous. But then after you normalize, it's like you have zeros in one hand and all the other values on the other hand. And this is an issue, of course, when you try to model the values of gene expression and variants and so on. Great. Anne has overtaken the first place. Let's move on to the next question. How many HGV shall we select for downstream analysis? So I encourage you to look at all the options carefully. How many highly variable genes shall we select for downstream analysis? Which one of these do you consider? Yes. This one is a tricky one. And I guess to some extent, not 100% fair, because I did put in the slides that between 2,000 to 5,000 is what people select. And it's also what most people chose. However, I also made the point that it's better to try different values. And this is the right answer, because I don't want you guys to have this pretty much this random value in your head, and because they said 2 to 5 is good, so I'll do 2 to 5. When the real message we're trying to teach is that, because this is an arbitrary value, it's good that you try different ones. So let's move on. Okay, now we have another top scorer, Anna. Question number 6, what is the effect of data scaling? Data scaling, if you remember, is our second type of transformation that we perform on our data set right before PCA. Yes, so most people got it right. Everybody got it right. I guess some people didn't have enough time. I thought we had 10 players. But anyway, the right answer is that it makes all genes have similar value distributions. So when you scale in the data, is a common practice in statistics. And the whole point is that you move the average for all your values to 0, and then you also move the standard deviation to 1. So that way you can compare across features and you can make direct comparisons. If you don't scale it, well, in our case, the genes that have higher values are going to dominate over the genes that have very low values. Let's move on to the next question. Okay, I'm taking back the first spot. See, what is the best description of a metagene? This has to do with PCA. Yeah, I guess that's all what I should say about this. Great. Yeah, most people got it right. It is a one-dimensional feature that captures the variation in gene expression. So one way that you can think of it is that instead of looking at the value of 1,000 genes, you can look at the value of one metagene that captures those 1,000 values. And we went over a little bit of the details of what it means to capture all those values. But yeah, that's the idea. And I'm glad most people remember. Great. Let's move on. Can zeros be captured in PCA? I guess this one is a tricky one, but it was mentioned in the slides. And it is something very important to consider when doing your data analysis. So when we perform PCA, what happens? Oh, I see. Only one people got it right. One person got it right. Yeah, so the right answer is that yes, zeros are captured in PCA, and in fact, they correlate with PC1. So what that means is that if you remember the definition of PCA and what the different PCs represent, PC1 is the dimension, or sorry, the axis that captures most of the original information, so most of the variance. And I showed a plot of one study where they correlated the values in PC1 with the fraction of zeros. And there was a pretty good correlation. This means that the signal, the variance that PC1 is capturing is mostly due to the amount of zeros, is coming from the zeros in your distribution. So that's why it's important to select a good number of PCs so you can capture all the biological signal and not just the zeros. So yeah, I guess this one was a tricky one. Let's move on. Since it remains unchanged, let's move on to the next question. What algorithm is used to find the neighbors of a cell in the PC space? Yeah, so this was also mentioned. We mentioned, I believe, all these algorithms yesterday, but only one of these deals with finding the neighbors for every cell. Yeah, people got it right. The KNN. Actually, the NN stands for nearest neighbors, so that's a little cheat. And K is the user-defined value, like how many neighbors do you want to find. Some people select a UMAP, and it's not 100% wrong, because the explanation of UMAP is dimension reduction, but I did emphasize the fact that it maintains the structure of the manifold of your data and the vicinity of the points that were, so the points that were close in high dimensional space. So, however, UMAP itself is not really finding neighbors, right, for every individual cell. UMAP is the way to represent it in 2 dimensions. And the neighbors and the clusters were defined, the neighbors were defined with this algorithm, and the clusters were defined with the Louvain algorithm. UMAP kind of receives information, the input, the output from these algorithms in order to perform the manifold approximation, meaning the 2-dimensional plot. Okay, moving to the next one. Last question, what is a marker gene? Hopefully this one is clear. This one we did cover today, so should be fresh in people's memory. Right, yeah, that's the right answer. A gene whose expression helps identify clusters. That's the very short definition, but that's the idea. This answer, a gene that is expressed in multiple clusters, that is a requirement also. Like we mentioned, it should be present, right, in other clusters in order to be testable. But that fact alone doesn't make it a marker gene. So, great. Okay, we have Anna in 3rd place, Jenny 2nd, and Anne. Were these the same winners as yesterday? I think I remember Anne and Anna also got the podium yesterday. But anyway, congrats, and you all win a round of applause by everybody else. Round of applause by everybody else. Okay, nobody wants to applaud. Well, at least you won. Moving on to the slides. Okay, now we can move on into day 3 material. And before we do that, is there any questions? Okay. Right, so the plan for day 3 was to continue with the clustering, which we sort of did by going through studying the marker genes and pretty much how they are the labels for every cluster. So we're going to start with pseudo time and then move on to some perspectives and future directions. So let's talk about pseudo time. Pseudo time is a type of analysis that we can perform on single cell RNA sequencing data. And the goal of this analysis is to reconstruct the path of cell differentiation based on our data. So what that means is that if you're familiar with the process of cell differentiation, we'll usually start with a type of cell, a stem cell, or a cell that has the ability to differentiate. And this cell will pretty much convert itself into a new type, or it can differentiate into separate types, causing a branching. And eventually, in this particular case, move on all the way to adult cancer or somatically different cells, differentiated cells. So if you think about this process, it's a process that is taking along throughout time, meaning that at a certain time we had our totipotent cell, meaning our paternal cell line, our stem cells that is to be differentiated. And then later on, as we move in time, we're going to get a different cell type and so on. So it's something that's taking place over time. But if you think about what single cell RNA sequencing is, it's kind of like a snapshot of the transcriptome, right? We're collecting the RNAs that were present in a cell in a given time. So we don't really have this time access in single cell RNA sequencing, but what people figure is that based on their gene expression profile, you can sort of figure out at which time step each cell was present. So that's why it's called pseudo-time. It's not a real time analysis. We're trying to figure out the time point based on the expression of our cells. So the process of pseudo-time is making some assumptions on the data. And the most important assumption, of course, is that the development of cells can be modeled using a smooth continuous surface. The way you can understand this is that the pseudo-time algorithm is actually assuming that in the process of cells differentiating, changing into a new type of cell, this process is taking place in a kind of like steady state velocity. So if it branches off, for example, not one trajectory is too fast compared to the other one. And it's also taking point in this one-dimensional space, or in this one-dimensional area that is time. So it's only moving throughout time. This is probably not super intuitive at the time. Oh, sorry, super intuitive at the moment, but it's in terms of the shape of the distribution, it becomes more clear what this assumption is talking about. And the second assumption, of course, is that the cells are undergoing transition. So how we motivated pseudo-time analysis is that we're trying to figure out how these cells go from the pluripotent state or from the stem cell state to the more differentiated cells, right? So we care about that trajectory. However, in the sample that you're analyzing, in all of the cells that you have, maybe some of them are not part of that transition. Some of those cells may not have nothing to do with that transition. So, and that's actually a big issue when it comes to pseudo-time algorithms. And there is a lot of effort into trying to figure out proper ways to include relevant cells only. And this third point kind of relates to the initial point that I mentioned, and is that because we're only looking at time in a one-dimensional space, we can't really account for complex time, say one branch differentiating much faster than another one, or differentiating and I'm going to say cycling back, but there's actually ways to deal with that, but it's a little more sophisticated. So what this assumption is getting at is that because we're looking into a one-dimensional projection of our data, or one smooth surface, actually, I shouldn't say one-dimensional, one smooth surface of our data, we can really consider very complex transitions. So here's a little overview of what this analysis looks like. And this figure was actually extracted from a 2016 paper, which is the one that introduced this idea of pseudo time analysis. So as you can see in this first panel, you see all these axes representing that all of our data points are plotted in a multidimensional space. And so the motivation was that in this multidimensional space, we can come up with a surface, a projection of these points that represents this time step transition into going from one type of cell into another type of cell. So I think at this point it's good to clearly establish the differences of dimensionality reduction and pseudo time analysis. Because these are different things, but just because we're talking about multiple dimensions, it may be confusing. So with PCA and UMAP and TSNE, which is another method that we didn't go through, but it's also dimensionality reduction. These methods are just trying to find the axis by which you can best represent the original data in fewer dimensions. So that's dimensionality reduction. Pseudo time, on the other hand, is trying to find the manifold, or like the topology, the structure in this high dimensional space that that kind of explains, that kind of shows a trajectory of the evolution of the transition of this cellular differentiation. And once you have figured out the manifold, the next step is to construct a transition matrix, which there are statistical methods to try to figure out the transitions going from point X to Y or to Z and assign them a weight, a probability. And finally, based on this transition matrix, this graph structure that you generate, the algorithm will try to figure out what's the best path throughout the data points. What path makes the most sense based on these ages, based on the overall shape of your data set. And eventually, it will perform this branching point identification. So what you need to take away from this plot is that once you have figured out the trajectory and how different cells connect to each other, one very important thing you want to figure out is what kind of transition is taking place. Is it a linear transition? So that will be a straight line going from one type of cell into another one. Or is there some branching going on? So starting from your original cell population starts differentiating into two or more different types of cells. So this branching analysis is also determined by the software that you're using, and is doing so by analyzing this trajectory graph in the first place. So just to give more of a clear example of this pseudo time, this is an example to put together in a review paper where it's pretty much comparing the performance of two approaches used for pseudo time estimation, T-scan and diffusion maps. But the idea that I want to convey with this plot is that here the y-axis is showing the differentiation of fertilized egg that converts into a zygote and then starts differentiating, multiplying into the state of 2 cells, 4 cells, 8 cells, 16 cells, and so on. So in our traditional single cell RNA analysis, we'll usually obtain the expression profiles for these different cells, try to reduce the number of dimensions and then find clusters. And we'll obtain these similar clusters, but in a UMAP plot, right? And a lot of times, you know, from the UMI plot, you may start to see this sort of trajectory, how this zygote, for example, seems to be closer to the 2 cells, and the 2 cells is closer to the 4 cells, and so on. And a lot of times it's not very obvious from the UMAP plot, but we can find one manifold, we can find one layer like we saw, or one plane in our high dimensional structure, where this pattern is going to become a lot more obvious, a lot more continuous. And in this case, our pseudo time, the units, is pretty much the coordinates in that manifold that we found. And in this particular case, and also by looking at how the expression of the genes changes from cluster to cluster, we can infer a trajectory that will connect these different cellular cell populations. So at this point, this pseudo time trajectory is not part of our tutorial, but nevertheless, I wanted to show you how, sorry, here. On this very same package, Surat, that we're using, we can perform this pseudo type analysis. So starting from this place, this plot where we identify, right, our two-dimensional clustering of cells using UMAP, and then later on we assign labels to every cluster. From this point, what is going on in this tutorial is that based on these labels, so we're selecting 2 types of cells, selecting erythroid cells and lymphoid cells. So we're pretty much splitting our original data set into erythroid and lymphoids. And now we want to build a trajectory between the cells that were clustered into these groups. And the way to do that, there will be some sort of data structure transformation steps over here. And I just wanted to get to this plot. In this plot over here, you can see that they're showing the representation in the UMAP. So in this 2-dimensional clustering, but the trajectory algorithm that was run on this data was able to come up with a 3rd set of coordinates, being the pseudo-time coordinates. And here in this example, the pseudo-time coordinates is shown by the different colors that are on our data points. And based on how the expression of different genes transition from cluster to cluster, they're also able to come up with a trajectory. So it's probably not very clear, but you can see how some of these points are labeled as 1, 1, 2, and 2. So this is, it's not very clear. I think it's indicating, I think in this particular case, it's indicating a trajectory going from this first step up to the 2nd step. But usually this type of algorithms will figure out the path, and will also try to figure out the order by which you move across the path. Now, a little side note, because you're probably wondering, why is this not included in our tutorial? The reason for this is that this is Surat, but it's actually borrowing some packages from another software, which is called Monocle. So the pseudo-time analysis is run by Monocle, which is another suite of analysis tools for single-cell RNA sequencing data. And it's also very complete. But Monocle has one major issue, and that is really hard to install it. So for that reason, I didn't ask you to install Monocle. And I figure it will be very tedious to try to run monocall all together at the same time. So I chose to just show the results in this case. And either way, it's the same thing, because you can think of these are your cells, and if you run the code, you're gonna get this plot. So hopefully we're not losing too much of the actual demo. Is there any question so far? Okay. Now, I wanted to move on to other types of analysis. So I believe with this, we have covered all, yeah, I would say all of the mainstream analysis of single cell RNA sequencing. And at least the analysis that you see more commonly in publications and what people are doing in order to uncover some cellular states, cellular transitions, and cellular heterogeneity. So, but there is also a lot of other methods, a lot of other types of analysis that are out there. And I think I mentioned before, this field is moving so quickly that every year there is so many, even like up to 100 tools that are being published. Like my tool does this, my tool does this other thing. So it's virtually impossible to keep up with everything. So I just wanted to very quickly mention what other types of analysis people are doing, and just so you have an idea and you know that these things exist. The first one, and this is also going back to, I believe somebody asked about this in the first day of our workshop about data integration. So data integration is actually still a very common type of analysis in single cell RNA sequencing. And I put it in the section of other analysis because it has to do more with the design of your experiment. And there are new applications that I'll mention later also. But the main idea, and the idea I want to get to first, is that this is a technique used when you want to combine together data from different samples. Now, these samples may be different replicates, or it may be, you know, cells from a different person or a different patient that you want to merge together into the same data set. And this is also an active area of research, because there is also a method in Surat that performs data integration. So we're just going to go over the basics and the motivation, and I can show you the Surat command to do this. So the idea is that you start with a reference and a query data set. So you see on your reference, you start with your two-dimensional clustering. You already identify the different cellular clusters. And then you also have clusters from a different sample. And you ask the question, a cluster in this data set, in this sample, how does it compare to a cluster in this other sample, right? So which one belongs to which? And the way to perform this analysis, very, very roughly speaking, is first, it has to be, you have to establish a unit of distance between the two data sets. In this case, an L2 norm distance, which is calculating based on the different gene expression values, how similar are the cells between one study or one sample to the other one. And then based on this distance analysis, you can sort of obtain a new coordinate system, a new set of values that will alter your clustering for both of your data sets. The way you can think of this is that a cell over here, I calculate the distance of this cell to all other cells over here. And it's a cell over here, it's actually closest to a cell over here, right? So we can come up with a new measure of expression that makes it, that reflects the similarity between the two cells. And then when you perform this, you obtain a new set of clustering where you see the coordinates, the position of the clusters actually is a lot more similar now between your reference and your query. And the third step is to find this set of anchors, which is once you have put them in a similar coordinate system, you try to find these anchor points, which are these cells that tend to reflect a very similar gene expression profile between the two samples. And so using these anchors, you can match one cluster on sample 1 to a cluster with sample 2. And as you can see, hopefully you will get a 1-to-1 match, but it's not always the case. Sometimes you can get what are considered two different clusters in the reference is actually considered only one cluster in the query dataset. Or maybe a cluster may not be present at all in the query, like the distance is too big to any other existing cluster. Yeah, so I think this is enough to motivate. It's probably not necessary to go through all these details, but I also wanted to show you how Surat has the option to integrate different data sets. And in this tutorial, they're actually starting from, starting from here. They're starting from one data set and then splitting it into two different ones. So you have two samples. And here you have the functions to perform the integration. These steps are pretty much the same that we perform. We normalize our data and we find our top 2,000 most variable genes. And then later on, here you see this function that is the find integration anchors. This refers to this point where we're trying to find the set of cells that will be more similar across the different data sets. And eventually here in this plot, you see that in this UMAP plot, you have cells identified from your original data set was split into this control and stim labels. And so here is showing how after integration, you observe pretty good overlap between the two data sets. So meaning you have successfully merged this information together. Is there any questions so far? Okay. Another type of analysis, this one is less common and is also it's also still like in the developmental stage, I would say. And the reason why I'm saying this is that there are a few studies that use RNA velocity, and there is also a few published methods, but it's still not this idea of a benchmark of which one is what's the best approach, what's the best way of going about this. It's not something that people have come up with yet. So, but just to briefly overview the concept of RNA velocity, I guess I should start by asking how many of you are familiar with the concept of splicing, RNA splicing? Can we do a show of hands? Okay, we have a few people. Very briefly, splicing refers to this this step that takes place in in the processing of RNA. And the idea is that you're cutting off pieces of RNA and then merging other pieces together. So you're really altering the original molecule. And the way this appears in our sequencing data is that some reads are going to be labeled as spliced and some reads are going to be labeled as unspliced. So what that means is that some reads will contain information about the original molecule, and those will be the unspliced reads actually. So they will contain information about the original RNA molecule, and the spliced reads will contain information about the modified RNA molecule. So what RNA velocity, what this approach really tries to achieve is to sort of quantify, sort of obtaining, just like in pseudotime, trying to obtain a trace, a path of the different cells based on the kinetics of splicing. So I think this figure is fairly technical. So hopefully I can capture the most important points. Here in panel B, you have, this is a model of how your RNA molecule gets transcribed from the gene at a specific rate, and then you obtain some amount of your original unspliced RNA molecule. And then splicing takes place also at some rate, and then you have your spliced RNA molecule. This spliced RNA molecule is your mature RNA, and it will go on to produce a protein and everything else. And then later on, it will be degraded also at some rate. So here you have a sort of kinetic model about the life of an RNA molecule. And I think we can skip directly to this last three plots over here. So the idea is that based on this ratio, based on this proportion of spliced versus unspliced RNA molecules, you can also come up with a type of RNA metric. And so in this case, this study focused only on circadian genes. Circadian genes are those genes that have whose expression, whose activity, is strongly linked to the time of the day. So, of course, the circadian genes are going to have different signals at different times of the day. And what this RNA velocity analysis is trying to achieve is trying to find the pattern, like how the proportion of spliced versus unspliced is moving throughout these time coordinates. I guess to some extent similar to the pseudo-time idea, but I think one of the biggest differences is that here we're not looking at different cells. We're just trying to figure out how this processing step is taking place for a given population of cells. There's a question, I believe.
Oh, sorry, I didn't lower my hand.
Okay. Yeah, so as you can see, the study of RNA sequencing is not only limited to gene expression. You can also study other cool features such as splicing. And here, I also included in this section, this is not a new technology. This is just another consideration in general, which is batch effects. And this is also linked to the data integration part, which is how much of your signal is coming from biology, from the expression of genes, versus how much of your signal is coming from technical factors. In this particular case, we have data that was that the that had two different library protocols. We have the SmartSeq and the SmartSeq 2. And when we prepare data using different libraries, and then we sequence and obtain our feature count matrix and perform PCA, we can see that in the PCA, the data points coming from either protocol, they're actually kind of clustering together, right? And this is in the first two PC dimensions. So the way we interpret this, of course, is that a lot of the variance, or most of the variance that we're observing in our data is due to the protocol that we used. And of course, that's not something, that's something that's going to affect your study. And the way to approach this, there is a number of methods out there. And when we talk about data integration, the algorithm is also trying to figure out the effects of the batch on your data, before trying to match it to a new data set. And yeah, and in this case, this particular data set you see is deeply confounded. And so you'll probably end up clustering these cells as one cell type and these cells as another cell type. When in reality, they may not be different cell types. They just happen to be prepared under different protocols. Yeah, so I also wanted to talk about another consideration, which is a very important consideration, which is cost. So how much does it cost to sequence single cells, and how much money should I spend in order to get a good amount of data? So this study put together some numbers. They evaluated a number of sequencing technologies, library preparation technologies, and here there is a true positive rate and false discovery rate. So some metrics by which, so we're calculating the price as defined by this metric. So what amount of sensitivity, what amount of specificity do we want? Here, this column over here, this is showing cell per group. So I believe this is the number of cells that you'll be finding based on 1 million, 500,000, or no, sorry, 0.25 million, 500,000, and 1 million reads. So you see also that for different technologies, the amount of cells that you're getting for the same number of reads is going to be different. And over here, this is the library cost, the estimated library cost for 1 million reads. And you can see clearly that it varies a lot from one technology to another, ranging from 0.1 to up to 25 or 30 million, $30 per million reads. And yeah, this is something we mentioned before, SmartSeq being relatively more expensive. Unfortunately, we don't have 10x in this table, but I'll show data for the 10x in the next slides. And yeah, and here is minimal cost. This is assuming, so every technology has a different cost associated to it. This is assuming on average $5 per million reads. And so given this number of cells, how much money you'll need to spend to produce sequencing for all of them. Now, I think most of us are affiliated to UCLA. Is there somebody who's not from UCLA? I think there should be a few people. Okay, So, okay, we have one person. Right, so hopefully this, the comparison applies. Hopefully the magnitude, the value is similar. Here, UCLA, we have, oops, sorry. We have our facility that will perform library preparation and sequencing. And here's a list of their cost for different sequencing technologies and for different library constructions. So the type of data that we were working with is 10x single cell, 3'N gene expression. I think this is a good point to mention the fact that 10x doesn't just do RNA sequencing. You see it has these other type of library preparations that it performs, just ATAC-seq. But for our type of data, we see that the price per sample or internal rate is around $2,000. So for us, UCLA students will cost around this much, and the external rate is a little more expensive, $3,000. So whoever raised their hand who's not from UCLA, you're going to have to pay a little more. And this is for library preparation, and there is also this list of costs associated with the sequencing technology. So the platform that was used for our data set was NeXT-seq. I forget which type exactly, but this is referring to the length of the reads. So of course, depending on how long the reads are going to be, the price is going to be different. But if we pick, for example, NeXT-seq per N 150 cycles, so 150 bases long, high output, this is costing around $4,000. So yes, that's to give you an idea, of course, of how much does it cost to generate this data. And this is this was extracted from the TCGB facility, UCLA Health. This is the facility where they perform sequencing and library preparation. So the way it works is that you need to submit your own sample of cells. So for the case of the 10x library construction, you need resuspended, sorted, dissociated cells at a given in a given PBS solution. And I think there is an ideal concentration of the single cell capture, this many cells per microliter. And there are some other notes on the quality of your sample. I think here I wanted to also show, I actually downloaded one of the forms. TCGB stands for Technology Center for Genomics and Bioinformatics Service. And this is the type of form that you fill in where you specify what type of library you want. Let's zoom in. Oops, too much. Fill out some information, the type of library that you want them to prepare for you, the type of, do you want to get RNA or DNA, the type of technology for the library preparation, and the type of sequencer that you want to use. And yeah, I personally never filled one of these forms. I think all of the data sets I've worked with were publicly available or were provided by a collaborator or were sequenced by other people in my lab. So I can't go too much in detail about this form, but I just wanted to show you guys what it looks like to submit a form to UCLA. And lastly, the last thing I wanted to show is some future perspectives on the field. And what are some things that people are looking forward that are going to be very exciting with single cell. And I think the most important one, the one I really wanted to mention, is single cell spatial transcriptomics. So spatial transcriptomics is a new technology and was actually named Technology of the Year in the year, I forget if it's 2000 or 2020 or 2021. But spatial transcriptomics is this type of sequencing where I used this diagram before to show how you can find many different types of cells within a very small area of tissue. And this plot was actually from a review paper in spatial transcriptomics. And what it's showing is that what this technology is doing is not single cell at the moment, but it's performing bulk RNA sequencing, but it's including some information about what part of the tissue this sample came from. And this has become a very, a very important technology because now, researchers are being able to to map a given transcriptome profile to a specific part of the tissue, a specific physical location of the tissue of interest. And based on spatial transcriptomics, you can perform an array of analysis, one of them being cell type deconvolution, meaning based on this bulk analysis, trying to figure out what the composition of different cell populations, what the composition of different cell populations is. And as of now, I believe there has been some attempts, some very pioneering work regarding single cell spatial transcriptomics, but it's definitely still not in the mainstream. I say this with a little hesitancy because, like I said, there are so many methods developed, and the amount of new technologies that come every day is a little overwhelming. And as far as I'm concerned, as far as I know, single cell spatial transcriptomics is still not something widely available. And another thing that is very exciting is the single cell RNA-seq, single cell attack fusion. So very briefly, this also has to do with data integration. And it's time to integrate not just different samples, but actually data points from different technologies, they're targeting different biological features. So RNA sequencing mostly is used for gene expression analysis. So this type of technology is telling you about the transcriptome profile, like what genes are being expressed and how much. And attack-seq is actually showing you regions of the chromatin that are, these are regions of the chromatin that are kind of transcriptionally active. So that because in the inside the nucleus, the chromosomes, the more compacted they are, the less transcribed the genes are. Whereas if you have more loose chromatin, then that means there's a more active area of transcription. And that information is captured here with the attack, with the single cell attack experiment. And so just this week, actually, in one of the seminars, one professor was presenting the method, the approach that they developed in order to merge these two types of technologies. And of course, you expect that a region that is transcriptionally active, you'll observe also high levels of expression. And lastly, the third point that I put here is long-read single-cell RNA sequencing. So same as before, I believe there has been already a few papers that have tried this, and this included, but with major shortcomings, right? This example that I'm showing here, this is from a paper that did a targeted long-read single-cell RNA experiment. Now, if you're not familiar with what long-read means, it just means that, so far when we talk about sequencing, you kind of have to break the original RNA molecule into pieces in order to sequence it. That's the traditional approach, and this is due to the fact that if your molecule is too long, the sequencer will not be able to read it completely in its entirety. After a certain length, the error rate is going to start increasing a lot. There is a lifetime for the proteins being used. So there are a lot of technical limitations as to how long you can sequence. But a few years already is being around this technology that allows for the sequencing of the entire length of the RNA molecule as the so-called third generation sequencing, or just long read. And so far, it's being implemented on bulk RNA and DNA. And as far as I know, there's been some smaller attempts to do single cell long read RNA sequencing, but it's still something that is not in the mainstream. It's still something that is not widely used. So it'll be also really interesting to have this information for individual cells. Is there any questions so far? Okay, and with this, I wanted to conclude this workshop. I wanted to say thank you to all of you for attending and participating. Keep an eye for follow-up emails. I've told a number of you have asked me about the material. So I will make everything available, the recordings, the data set, the code. And I'll even include 1 or 2 review papers that I strongly relied on to make this workshop, and which I think will be a big help also. Just a very good reference to have when you're trying to learn about single-cell RNA sequencing. Those of you that are taking this course for credits, there will be a homework assignment and quiz sent by email later today. So yeah, keep an eye on the follow-up emails. And you'll have 2 weeks to complete the assignment, which it should be plenty of time. And if you have questions regarding the assignment or regarding the quiz, you can contact me by email. And those of you that are not taking this workshop for credit and still want to attempt the homework, also shoot me an email and I'll send you the homework to you. I'll gladly give you some feedback. Yeah, like I said, all the course material will be made available. And this part, I haven't heard from the collaboratory about this recently, but I think they usually have a survey for every workshop. So they'll ask your opinion about this particular workshop, and whatever your feedback is, is very valuable, because we read this, we read all these surveys, and based on that, we try to design the program for the next year or the next quarter. So whether you liked this workshop or you disliked it, please make sure to write some comments so we can work on it. And even if you thought there is nothing to change and it's perfect, then you can write that as well. Just write, it's perfect, don't change it. And yeah, and I just wanted to leave you with this very last slide, which is This is just a comic kind of pointing out the overused titles or like the common theme in research papers regarding single cell RNA sequencing data. And the one I like the most is this one over here. This paper that is titled, "No, it's not suspicious that our benchmarking study highlighted that our method is the best." And I find it funny because, you know, this is so common. When people introduce a new method, the way you want to present it to the rest of the community is by benchmarking, so comparing it to existing methods. And you know, it's a good practice, of course, but it's kind of a little fruitless because, you know, you're introducing a new method. Of course, it should outperform everybody else's. Otherwise, it wouldn't get published. So yeah, that's it for the workshop. Please let me know if you have any questions. I believe all of you have my email, so feel free to reach out via email. And like I said, keep an eye for any follow-up email that I'll send you, especially those of you taking the course for credits. Is there any question right now? Okay, if not, then that's it. Thank you, everybody, and feel free to log off.